Characterization is a very important step once it's been possible to purify proteins, or even during the process of purifying proteins, so that you can track what's going on. That means that you have to detect proteins. And the most common feature of proteins is their absorbance at 280 nanometers. That's because they contain tryptophan W, tyrosine Y, and phenylalanine F, all of which are aromatic amino acids that absorb in the range of 280 nanometers. And so if you have a protein and you look at its spectrum and it doesn't, and you're trying to purify it, and so you have a sample and you look at its UV spectrum and it doesn't have a maximum at 280, then you need to really think about what you're purifying and what's going on. You can also use fluorescence, which is primarily tryptophan. There are also agents that bind proteins. They're dyes like Kumasi blue, shown here, or silver, which is more expensive but also more sensitive. And we'll see examples of where these are used shortly. You can also use specific antibodies. You can use polyclonal antibodies to detect where a protein is, but even more specific are monoclonal antibodies that react with one specific antigenic site within a protein. And we'll talk about some interesting uses of antibodies. But how do you make monoclonal antibodies? And what might they be used for? And the way we make these is by a process known as making hybridomas. And the way that happens is that we start with the mouse and you immunize it with your antigen X. And the B cells, a portion of them will be making that antibody, and the rest of them are making antibodies to something else. And so you can collect the B lymphocytes from the mouse, but you can't separate your cells from the other cells, and the B lymphocytes do not survive but maybe a few days in cell culture. So that's not a very effective way of getting monoclonal antibodies. But there's a mutant line that's derived from a B lymphocyte tumor that's found in mice. And these cells will grow indefinitely in normal medium, but they do not survive in a selective medium. They have specific needs, and if you leave out specific components, they will not survive. So if you take two cells, there are compounds that promote fusion of the cells. And so you will get a subset that form these hybrid cells that are called hybridomas. They from the lymphoma side, and hybrid because they're two types of cells. And a subset of these, and you can see the little red and blue and yellow cell in the right set of cells, that's the cell that you're after because it makes the antibody you want. It's a hybridoma, and so it will survive and grow and secrete antibodies. So how do you get that one little cell that's in the midst of all these other cells? Well, if you let the cells culture the cells for a few days, and you culture in the medium that's toxic for the lymphoma cells, then all you get is hybrid cells. And a subset of those are going to be producing the antibody that you want. And so you can see that everybody dies out except the hybridoma cells, and then there are these cells in that right dish that you actually want. But how do you know that you want them? Because they're mixed in with other hybridoma cells. Well, what you can do is take the cells and plate them at a density that's 1 cell or less per cell, so that per well. And so that in any given well, if your antibody is being produced, you know that the cells there are the hybridomas that are making the antibody that you want. And there'll be a subset of those wells. You can take those cells and test them for the antibody that you want. And when you get the positive results, then you can take that original clone and grow it up repeatedly to provide a continuing source of a highly specific antibody. So hybridomas are very useful for producing very specific antibodies that react with a single antigen within a protein. Remember that there's a lot of surface on a protein, and antibodies are made to different parts of that surface. In this case, you're isolating a single point on the surface as a mode of detection. So what can you do with these? Well, we're going to set that aside for a minute and then talk about how you separate proteins in a mixture and how you can actually track purification. And this is a really important part of the process because you have to know, have you managed with any given step, ion exchange chromatography, gel filtration, different types of ion exchange chromatography, have you managed to purify your protein? And the historic article by Weber and Osborne describes SDS polyacrylamide gel electrophoresis. Sodium dodecyl sulfate is SDS. It is a detergent. It has a long aliphatic chain and a sodium ion attached to it, so that it is sort of like those membrane, the components of membranes that we've talked about that are apolar on one end and polar on the other. It turns out that SDS, the side chain, interacts with the backbone of the protein and the side chains, and that sodium at the end is charged, and there is a relatively common density with which SDS binds to proteins, and so you've got lots of negative sulfates that repel one another and stretch the protein out, in addition to the denaturing effect of the aliphatic chain. And it turns out that SDS gel electrophoresis works on mixtures, it works on purified proteins, it works on any protein. And that because you get this uniform density of SDS, and SDS is uniformly negatively charged, that there is a common charge mass ratio generated by SDS bindings, which allows the separation in the electrophoretic field by the length of the protein, by the monomer molecular weight, which is proportional to the length of the protein. You can detect the separation by dyes. Kumasi blue is shown here on the left, and the silver, which is more sensitive, but a little more complex to do and definitely more expensive, is shown on the right. So you can then see basically the proteins that you have in your mixture. And you do this in a plastic encased system that you can actually build yourself or you can buy. And this system is to support the gel and to separate the cathode chamber from the anode chamber. So the gel and the buffer within it is the separation, the continuity between the cathode and anode, so that you have an electric current that runs through the gel itself. And you place your samples in the sample wells across the top of the gel, and you load it with a little micropipette or a micropipette tip. And the samples can be different in different wells, and you can have as many as generally 20 wells across a gel. It's just easier to depict with 4. And you have your buffer at the cathode and their buffer at the anode. And you'll note that the cathode is at the end where you're loading your sample, because with SDS, polyacrylamide gel electrophoresis, your sample has been covered with SDS, and SDS is what kind of charge? It's negatively charged, and so you have a very highly negatively charged, now extended because the charge repulsion stretches it out, sample near the cathode, and so it will be repelled from the cathode and move toward the anode. And so here is an illustration of what happens with SDS. Sometimes proteins are linked by disulfide linkages, and so SDS gels are almost always run with SDS and mercaptoethanol or dithiothreatol to break up the disulfide bond and convert these to sulfhydryl groups. So you're always looking at monomer molecular weight. If you have a higher order oligomer, SDS breaks it up into monomers and unfolds the monomers. You're looking at denatured monomeric proteins with SDS gel electrophoresis unless you vary the conditions in some way. And again, they're highly negatively charged because of the SDS binding, and their charge mass ratio is uniform, so that when you separate these molecules, they are separated based on their molecular weight. And on the left here, you see molecular weight standards. So you can read basically the molecular weights of the proteins in the sample. And you can see here, if you look at this band, this very heavy band on the right, and trace it back to the left, you can see that you've managed to purify a protein. You can still see the contaminants in the background. This is adjusted so the contaminants are at pretty much the same level, but you've purified the protein against those contaminants. So it's a really good way to assess purification of proteins, and also to determine monomer molecular weight very easily with small amounts of material. You can actually do this in 2 dimensions, because it turns out that you can do a native gel in 1 dimension that is a pH gradient from basic on the left to acidic on the right. Proteins move based on their charge, and some proteins have lots of acidic residues, some have lots of basic residues, some are sort of in the middle. And if you kind of look at the distribution below from left to right at different levels, you'll see that there are proteins that are on the basic side and proteins that are on the acidic side. Because ions dissociate based on the pH, there will be a point at which the number of negative charges equals the number of positive charges, and the overall charge on the protein is 0. And that is the isoelectric point for the protein. And so as proteins move through this, they will move to a place where their charge is 0, and then they won't move further. And if they happen to ionize one way or the other, they'll move a little bit and then they move back. And so it's called isoelectric focusing. And so you get a focus of bands at particular pHs. So that's 1 dimension. And then if you take that gel and lay it on top of an SDS gel, and run it in the 2nd dimension, which is now down, now you have proteins that are denatured and monomeric. And so you can get an idea of both isoelectric point and monomer molecular weight. So why is SDS gel electrophoresis so useful in protein purification and for protein characterization? But what feature of a protein does it not measure?